Good morning and good afternoon to those on the East Coast, and good morning to those on the West Coast. Thank you for joining us here today. My name is Jessica Lachey. I am the marketing manager here at NanoScience Instruments. I have the pleasure of having Tom Hayes with me, the application scientist from Bionesia, who will be giving a presentation today on the fluid attack, as well as Matt Jobbins, who is our application scientist at NanoScience Instruments. I'm going to give people one more minute to get logged in. I know that if you are new to the GoToWebinar, it does take a little bit to download, so I ask for just a little bit more patience. You are, as an attendee, you are muted, so I will walk you through how you can interact with us during this presentation. So for today's agenda, I'm going to walk you through how to ask questions in the GoToWebinar interface. We would like this webinar to be an interactive one, so I will introduce to you how we're going to be interacting with you during the webinar. I'm going to take you through an introduction of NanoScience Instruments, the upcoming shows and events that we will be attending, and then I will be handing the presentation off to Tom Hayes, our application colleague at Bionesia, who will take you through a background of the Bionesia and the fluid Natech electrospinning and electrospraying equipment. And then Tom will be handing the presentation off to Matt, who will walk you through the phenom scanning electron microscope and how you can use this instrumentation in your research. So here I am Jessica Lachey again, marketing manager here at NanoScience Instruments. In your GoToWebinar interface, you'll see an orange arrow here. This is how you open and close the dialog panel. If at any time during the webinar you have a question for us, there are two ways that you can ask your question. You can raise your hand in the chat window here, and this will open up the chat window that you can ask a question for the whole group to see your question. Or if you would prefer to ask a question privately to us, you can enter your question here. And myself or David Marchand, who is our other application scientist at NanoScience Instruments, will be responding to you as best we can during the presentation. We will also be conducting poll questions during the course of the presentation. So NanoScience Instruments, if you're not familiar with who we are celebrating 15 years this Saturday of being in business, celebrating the scientific community. We have application-specific support for a wide variety of instrumentation. Today we're presenting to you the manufacturing line that we have for electrospinning and electrospraying nanoparticles and fibers. And then we're also presenting to you the desktop scanning electron microscopy line that we have. We also have mechanical testing tools. So if any of these are of interest to you, please contact us at applications@nanoscience.com. We have an applications team of 6 individuals covering both the East Coast and the West Coast. Our East Coast office is in DC, and we have Matt, David, and Nick. In our Phoenix office, where I'm located, we have Zach, Nicholas, and Dev. Each of these scientists have specialized application background and support each of the tools that we sell. The upcoming shows and events that NanoScience Instruments will be presenting at will be the MS&T booth at Pittsburgh, the filtration booth in Chicago, and the BMES in Phoenix. These are all happening next month, so if any of these overlap with your schedule, we would love to see you. Please come out and see us in person. At the filtration booth and at the BMES booth, we will be featuring both the fluid Natech and the phenom. So without further ado, I'm going to be getting ready to pass this off to Tom. Tom's going to be talking to you about Bionesia, giving you an introduction to the fluid Natech, understanding how electrohydrodynamic atomization works, production of nanofibers and particles, the control and process parameters that make the fluid Natech unique to electrospinning and electrospraying, and then he will be taking you through some application examples. After that, he will be passing this off to Matt, who will be walking you through the phenom and the unique design and how it can be used to quantitatively characterize the particles and fibers created from the FluidNet. Tom?
Hello, okay, thank you. Thanks everyone for registering your interest in this. So let's get started. Okay, so BioInicia is a private company. We are headquartered in Valencia in Spain, and we're specialists in the fabrication of nano and micro fibers and particles by electrohydrodynamic processing, so commonly known as electrospinning or electrospraying. So in Valencia, we've got our equipment manufacturing workshop. We've got R&D facilities based in one of the research institutes in Spain, from which we spun out in 2009. We've also got pilot plant equipment for product development and a manufacturing plant inside the clean room, which is pictured there, for pharmaceutical and biomedical device manufacture. So that's specifically for nanofiber and microfiber manufacture. We also have a production facility next door to this one for the manufacture of nanoparticles and powders. So we recently achieved ISO 13485 certification, which allows us to manufacture medical devices, and GMP pharma certification for drug-loaded devices. Okay, so we have three lines of business at BioInicia. The first is the FluidNet equipment range, which is a range of electrospinning and electrospraying equipment covering laboratory kind of benchtop equipment up to what we'd class as pilot scale or pre-production equipment. Then we have services for contract R&D, so developing products on behalf of our clients, and we have particular expertise in development of products for biomedical, pharmaceutical, cosmetic, functional food and active packaging. So products where we're applying some additional functionality to materials, and often in application areas requiring some good level of control and perhaps with regulations over their manufacture, such as GMP. And then we have contract materials manufacturing services using our own electrospinning equipment in the clean room facility. And for this, we can scale up products that our customers have developed in the lab, or we can take on the scale up from the contract R&D projects that we've done on behalf of our clients. We don't develop any products for ourselves, so we don't compete with our customers. We purely offer the services of development and manufacture. So just to introduce the technology, I guess some of you may be familiar with electrospinning and electrospraying, some of you may not be. So just briefly, our FluidNet equipment uses electrospraying and electrospinning, and this is a voltage-driven fabrication technology. So rather than using mechanical forces to draw fibers, we're using high voltage and electrostatic fields to stretch a spinning jet into nano or microfiber or particles. And the result is a dry product. So we start off with a liquid and we end up with a dry product. So the same process can be used as a drying method. Importantly, it's a room temperature process, which means that we can process materials which are particularly sensitive to thermal degradation or oxidation, and even living cells. It's a process that can be used for a very wide range of materials, which means that it's got a huge range of possible applications. And the materials can be natural polymers or proteins, synthetic polymers, ceramics, carbon, all sorts of things. And we can also, using this technology, produce a range of different complexities of the actual structure of the material. So we can create simple fibers and simple particles, or we can process through coaxial spinning heads to produce core shell structures, for example. And this is particularly interesting for encapsulation of functional materials. critically, this process that we use in our Fluidinotech equipment in our manufacturing line allows for tight control of fiber and particle dimensions and stability over time. And it's a scalable process, so we can increase the number of spinning heads, increase the size of the machines, and thereby increase the production rates up to industrial scale. And this we've demonstrated in our own plant. So this is just a diagram of electrospinning setup in its most basic form. So we start off with a polymer or a protein or whatever material you want to produce. You start with dissolving that into a solution or creating a suspension or an emulsion. And we pump that through a capillary needle spinning head, usually using a syringe pump at lab scale or other pumping methods at industrial scale. And then we apply a high voltage to the electrospinning needle. And as the voltage increases, electrostatic repulsive forces experienced there by the droplet of solution at the tip of the needle. And the droplet changes shape into what we call a Taylor cone, the image on the left-hand side there. And from the tip of that Taylor cone, a solution jet is expelled from that droplet, and the jet undergoes interactions with the electrostatic field, which cause whipping instability, and that whipping stretches the jet at the same time as solvent is evaporating from the solution from the jet, and eventually the dry product is collected on a grounded or negatively charged collector. But on the right hand side, this is a high-speed camera shot of the spinning jet, and you can see that it's one continuous fiber which is collected per spinning needle. And obviously we use many needles to scale up the process. Electrospinning and electrospraying are very similar in processes, and the hardware is the same. So our equipment can be used for both. We can manufacture fibers and particles in the same machines, the difference being the solution that we process. So a higher viscosity solution which has molecules inside which entangle with each other during the stretching of the spinning jet results in fiber formation. A lower viscosity solution which doesn't have the same entanglement within the solution as it's spinning, it means that the solution is able to break up into droplets, and those droplets break up further into smaller and smaller droplets, and you end up creating nanoparticles. And in both cases, the range of diameters is well controlled, somewhere between about 50 nanometers and several microns, with a typical range for fibers being in the hundreds of nanometer diameter range. Okay, this just shows some of the key parameters involved in the electrospinning technology. So on the solution side, we've got viscosity, which is controlled by your recipe, your polymer concentration and molecular weight, the conductivity, surface tension, and the solvent volatility. So those are all aspects which are designed into the process through your solution recipe. Then there's the processing parameters, voltage and distance, which influence the electrostatic field strength, and the flow rate. And in the case of spraying particles, flow rate is a key parameter in the control of particle size. In the case of fiber formation, and viscosity is the key parameter. And importantly, there are environmental parameters which have a significant effect. So temperature and relative humidity in particular can influence the evaporation of the solvent from the spinning jet, and therefore the resulting morphology of the fibers and the particles. So in order to keep stable, consistent product morphology, you need to stabilize the environmental parameters. So using this needle-based electrospinning process means that we can obtain single-phase materials, simple fibers, and we can also create multi-phase materials, so either by blending of two or more materials into one solution, or we can process through it coaxial spinning heads, and that consists of one needle, capillary needle, inside another capillary needle, and you flow 2 different solutions through each one and end up with a core shell structure. And that can be, I mean, typically you'd have a core of a functional bioactive material which can be released over time, or you may want to have the functional coating on the outside to interact with its environment, for example, in sensing applications. Just to give an overview of a few of the materials involved in this process, we can process synthetic polymers. There's a very wide range of polymers published in literature that we can use as a starting point for development projects. So nylon is a very common one, PVDF, polystyrene, etc. Biopolymers, which Bionisia has particular experience with, are very useful for biomedical applications or food applications, for example. Encapsulated actives is often involved in the kind of projects that we do. So we can process functional and active materials, even if they're not spinnable themselves. We can blend them with spinnable polymers or proteins, or we can process them through core shell spinning. And then inorganic fibers is another important aspect for some applications, such as those that would be materials for use in high temperature applications. So we typically see titania, silica, alumina, zirconia fibers, and these are processed through a sol gel type process. So we spin fibers from a sol gel, and then they undergo a heat treatment to burn away all the organic fraction and leave the pure inorganic fibers. So compared to other processes, well, most textile manufacturing processes produce larger fibers, so typically more than 5 micron in diameter, or more often more than 10 microns in diameter. But there are some other competing technologies for creating nanofibers, for example, self-assembly, microbial fermentation, and free surface electrospinning, or nanospider electrospinning, and force spinning. But so some of the advantages of our electrospinning technology include a tight control of fiber or particle size. Because we process through capillary needles, the solution isn't exposed to the environment until it's actually spinning. So there's no uncontrolled solvent evaporation that happens in some of these other processes like free surface electrospinning. We can control the environmental conditions through climate conditioning. It's a room temperature process, so we don't need to worry about materials that are thermally sensitive or oxidize. We can process a very wide range of materials, which means the process is applicable for many different applications, and it's a scalable process. So by increasing the number of spinning heads, we can increase the productivity of the process. Whereas self-assembly is only really at lab scale. It's a bottom-up process which takes a long time. The microbial fermentation is very limited in terms of the materials you can produce, typically forms of cellulose. Free surface electrospinning, which has great scalability, but more of a limited selection in terms of materials. It's more limited for using volatile solvents because there's some uncontrolled solvent evaporation from the process, and more limited deposition options. So if you're wanting to coat complex shapes or creating tube structures or anything like this is much more difficult in that type of process. And force spinning, again, gives you less control. This is like a centrifugal spinning process, but it's difficult to control the fiber diameter, and the deposition options again are quite limited to kind of roll-to-roll applications.
Okay, here we have our questions. I don't have any questions that I see that have been asked and through the chat window or the question window, but I am going to do a poll for you. So how do you currently create nanofibers or particles? Are you using a home-built system, another commercial system, or you don't currently do this but you wanted to learn more and you want to learn more about the FluidNet-Tech? So most of you have said that you don't currently do this, and so this is a great opportunity for you. About a quarter of you are using a home-built system, and then about 20% of you are using other commercial systems. So we're happy to be able to share all of this with you today.
Okay, thank you. So just to introduce you to the range of electrospinning equipment that we sell. So we have the FluidNet-Tech LE10 on the left-hand side, which is our smallest machine as a laboratory benchtop equipment suited. We'll go through in more detail in the next slide. The LE50, which is a slightly more advanced machine, still a laboratory benchtop equipment, but able to work with climate conditioning. We have the LE100, which is a larger machine. We kind of target towards advanced product development and also in some cases manufacture. So we sell this equipment to some customers working with ISO 13485 or GMP certification for manufacture of their medical devices, for example. And the LE500, which is targeted towards scale-up pilot scale manufacture on roll-to-roll type applications. Crucially, this technology is scalable. It's transferable from one machine to another, so you can start your developments on a small benchtop system and then scale them up with the larger equipment, either the LE500 or LE100, or at Bionicia with the industrial equipment that we have. And it's flexible, so all the machines are modular, and we can configure them so that they can be tailored to your particular needs. And all of them give you good control over the products that you're manufacturing. And from the LE50 up in particular, the systems can be used with our climate conditioning to control temperature and humidity. So the LE10 is the most basic machine that we sell. It's able to produce fibers single phase or coaxial, to collect onto stationary plate collectors or rotating collectors if you want to align fibers or produce larger sheets of uniform material about the size of A4 paper. And it's a small system. It's very simple, good for people just setting out the process and learning about the process and characterizing materials. The LE50 then is still a very simple machine, but is crucially compatible with the temperature and humidity control. And it has a larger chamber, which allows you a bit more flexibility in terms of the apparatus set up inside. So in this machine, you can spin, for example, 2 fiber types at the same time, collecting on a rotating drum, so that you can create a composite type structure, for example. Both of these machines can be used with a multi-nozzle injector with 5 emitters. So for some slightly faster production, you're not having to wait quite so long as using a single needle apparatus that most people's homemade systems use. The LE100 has a few more options. So we have the ability to work with 3 solutions. So this could be, for example, 2 solutions working in a coaxial nozzle and then another solution being spun simultaneously. We can also move the spinning head in 3 dimensions. So we can create some 3-dimensional patterning, or we can coat larger objects, for example. This one works because it's a larger machine. We have some more space inside the chamber for additional apparatus. We can work with larger rotating drums, up to 200 millimeters diameter, which gives you a sheet of nanofiber approximately the size of A3 paper. and larger spinning head with 20 needles, so you can get you can produce your samples much more quickly. And this equipment has been really designed to be very easily cleanable, to be usable in clean rooms, and to offer very good control over all of the conditions. Everything can be data logged. It can be supplied with multi-user level access, so that you have a administrative login to change all the settings and set up the experiment, or an operator login where you can just select a pre-saved set of parameters, and they can be loaded in automatically to the machine. And the LE500 is really focused towards scale up of roll-to-roll type material. So we have an internal roll-to-roll collector, which is like a conveyor belt which passes a substrate through the spinning area. So we coat a substrate, and then that can be either a substrate where the fibers adhere or don't adhere, so you can remove the substrate and leave the pure nanofiber product if you want. This system works with pressurized solution delivery, which means that we can work with solution reservoirs which of larger volumes, or up to 2 liters, and you can have 2 of those working together. So you can process materials in much longer batches, and also it gives you the ability to spin from up to 96 needles simultaneously, so having much higher production rate than the laboratory scale spinning. So as I said, from the LE50 upwards, the equipment can be used with our climate control AC unit. So this controls the temperature and humidity inside the spinning chamber. And it does this by conditioning the air before it enters the chamber. So in the photograph, you can see the AC unit just to the right of the Fluidnatech LE500. And on the right-hand side, we see this section of chart showing the temperature humidity range, which is achievable using the AC unit in our electrospinning equipment. So, for example, at room temperature, so on the bottom, the x-axis, we see the temperature, and the curved red lines indicate the relative humidity, and the shaded blue area is the area which is achievable with our AC unit. So, for example, at 25 degrees, we can achieve relative humidity inside the chamber between about 30% and about 75%. And this control is particularly important when you're wanting to create consistent product. So very important in the manufacturing setting, particularly for medical or pharmaceutical type applications, but in many others as well. And it's important because temperature and humidity have a strong influence on the evaporation of the solvent, and therefore the spinning of the polymer solution and the resulting morphology of the fibers. So variations in the conditions in the chamber will lead to variations in the morphology of fibers, perhaps the diameters, and also in the formation of unwanted artifacts, such as beading along the fibers. So we can use the AC unit to either control the temperature and humidity to be very stable, so that you achieve consistency, and you can also use it as an experimental tool to optimize the spinning conditions. And as an example, here we had a project where the customer had done laboratory scale spinning of their polymer at room conditions, so 24 degrees and 35% humidity. And we were wanting to improve the fiber diameter variation and increase the productivity. And by optimizing the temperature and humidity, so increasing the temperature and reducing the humidity, we were able to achieve 4 times the manufacturing, the productivity, and reduce the diameter variation. And this was achievable through the control of the temperature and humidity.
Okay, now we have time for some questions. I do have quite a few questions that came through after that last break. For the first question, Tom, I have for you.
Fire away.
What is the material quantity range needed for the LE10?
So in terms of the materials, the raw materials used, we can work with only a few milliliters of a solution. Typical electrospinning solutions for fiber formation may be in the range of 5 to 20 weight% of solution, and we can work with as little as 1 milliliter if needed. We can use different sizes of syringe, so from very small ones for processing small quantities up to, I think, 140 milliliters is the largest syringe size that fits into the pump. So we can work with very small amounts. We also have an accessory, which is a tubeless solution feeding system. So rather than the typical setup, which pumps solution from the syringe through some Teflon tubing to the spinning head, we can connect the fluid reservoir directly to the spinning needle if dead volume is a concern.
The next question I have for you, does the needle capillary move during the spinning to enhance uniformity of the web?
Yes, so when we're collecting onto rotating drum collectors or mandrels, we typically move the spinning head, the needle, in one axis, so back and forth along the length of the drum. We can also, in the LE100, we can move the spinning nozzle in 2 or 3 dimensions, so we can coat uniformly a stationary flat collector as well.
And then what is the difference in process between free surface electrospinning and the fluid Natek?
So our process pumps a solution through capillary needles so that the solution is enclosed until it's spinning. The free surface electrospinning has it's an open surface, and the process started out as having an open bath of solution with the electrode was a rotating drum which picked up a thin layer of that solution, and the drum was charged, and spontaneously the jets of polymer solution form from anywhere they like on that surface. Now the process is developed to improve control slightly, by, rather than using a drum, they use a wire, and still the solution is just wetted onto the wire, and the spinning jets form spontaneously. So it doesn't give us as good a control. You're not ever able to directly control how many spinning jets are forming at one time. And also there's this uncontrolled evaporation of solvent from the solution. So you have a more limited range of solvents that you can work with, and you end up with a bit more non-uniformity over time as you're spinning.
Thank you, Tom. I'm going to take a pause on questions right now. I'm going to launch another poll. This will help us for future webinars. This is specifically for applications. For what webinars would you like to see in the future for applications? A tissue engineering cellular focused webinar, cosmetic focus, filters non-woven focus, or other? And if you could please let me know in the chat window for the other, so that I can take note of that, and then we will be setting up other webinars in the future for those topics. All right. Thank you, everybody, for your input. We have quite a few, we have a wide variety of interest, so I'm not going to share the poll results with you, but please stay tuned from NanoScience Instruments for upcoming webinars in the future on those topics. And now to our application section.
So in the interest of time, I won't stay in this section for very long, but just to give a brief summary of the various applications that our technology is used for. So in the biomedical space and the pharmaceutical space, I've got a couple of specific slides next. Energy So it's these kind of nanofiber membranes are used in battery and fuel cell separation membranes, in hydrogen storage applications, photovoltaics, transport. Electrospun nanofiber is showing some great results in improving the performance of carbon composite materials, and is able to also add some functionality, so either in self-healing composites or perhaps sensing composites. Filtration, well, this is probably the oldest application for electrospun nanofiber. By using electrospun fiber, you can create very small pore size, which means that you can capture smaller particles, and this has been used commercially for many years. Agrochemical industry is nanofibers and also nanoparticles are of interest for pesticide controlled delivery, for efficient use of chemicals, fertilizers and pesticides, and for plant protection. And in the functional textile space, we have the ability to make flexible electronics, so nanofiber-based electronics and sensing fabrics, to incorporate antimicrobial materials onto textiles, to use like the nanofiber membranes, like a Gore-Tex type waterproof membrane, and also for chemical protection suits, shown interest in nanofiber materials. So specifically, the pharmaceutical area, high surface area, it means that you can deliver very efficiently a drug or a bioactive. It's a room temperature process, so you don't degrade the bioactive materials, and that's particularly important for cosmetic applications, where a lot of the actives are oxidized very quickly. And there's flexibility over the design of the fiber or the particle. So by using this kind of core shell structure, you can design the release profile of your bioactive. And so this sees applications in the form of controlled delivery devices for drugs and bioactives. So we can release a bioactive in a sustained way over time, or we can achieve a multi-phase drug delivery profile by incorporating a drug into various layers of your structure, and even by selecting the materials, we can target the delivery to a particular area of the body, for example, into the intestines rather than the stomach, or vice versa. It's also used for the protection of degradable materials, so to protect them from oxidation and give a longer shelf life, you can encapsulate them using this electrospinning and electrospraying process. And also, interestingly, because it's a drying process and it dries the material very quickly, you can actually use electrospraying to control the level of crystallinity of a drug product, which means that you can produce a more amorphous drug, which increases its solubility in the body and bioavailability. So this is one case study. So this is a product that we developed at Bionisia on behalf of a pharmaceutical client, and we'll now be manufacturing this under our GMP certified manufacturing plant. And this is a 2-layer patch, a nanofiber-based patch, which is designed to release a drug into the oral mucosa, so the gums or the cheek. And this is a 2-layer product containing a hydrophilic layer containing the drug, and a hydrophobic layer which acts as a barrier to prevent the drug from releasing into the mouth. So all the drug is released through the mucous membrane. And in the biomedical space, which is another important application for the nanofibers, it's particularly important because the structure that electrospinning forms is biomimetic structure, so it creates very similar structures to the natural tissues that our body's made out of. It has a high surface area, which means we have a very high area of interaction with the body. So if we add any functional ingredients, for example, then those are very efficiently used. And we can control the fiber and the fiber diameter and pore size to make it to optimize the properties for particular applications. So in tissue regeneration, for example, you can create scaffolds for cells to grow on. And because the scaffolds mimic this kind of structure that the cells are used to growing on the body, it means that they behave more naturally. It's also possible to deliver living cells through the process, so you can simultaneously be spinning a fibrous scaffold at the same time as depositing cells into the scaffold, so that it's a pre-seeded structure, and you don't have to then find a way to seed cells into the center of the scaffold. And for wound healing, it's of interest because the nanofiber-based materials with small pore size and high surface area allows for moisture and air exchange through the membrane, but they protect the wound from wound infection from bacteria, and because of the small wound size, small pore size, sorry. And in implant coatings, again, this you can coat an implant with a nanofiber material which contains perhaps a growth factors or some other bioactive material to increase to improve the biointegration. So on the right hand side, we can see a bit of movement here. So this is heart cells which have been grown onto aligned nanofibers. Aligned nanofibers you produce by collecting onto a fast rotating collector. And because heart cells are used to growing on aligned collagen fibers in the body, it means that they're behaving more naturally. These are cells which are grown in the lab onto aligned nanofiber scaffold material, and they're spontaneously beating without any stimulus because they're behaving as they would in the body. On the left hand side, these are brain cancer cells, which in the body they would be growing along aligned collagen fiber. And so this laboratory experiment is conducted on aligned nanofiber, and the cancer cells are growing very quickly in the direction of the aligned fiber as they would do in the body. And this is important for example, drug discovery and drug testing applications in the lab, where you want the cells that you're testing your drugs on to be behaving as they would do in the body. And this is what we can achieve with our technology. Okay, and I think that concludes our just brief summary of applications that are possible.
Yes, thank you, Tom. We are running a bit long, so I apologize to everybody that had questions that we didn't get to. We will get to your questions offline later on. I'm going to be passing off the presentation now to Matt, who's going to be going into the desktop Phenom.
All right, thank you very much, Tom and Jessica, for the overview of the BioInesia fluid and tech electrospinning equipment. So as we, I guess as Jessica said early on in the talk, right, NanoScience Instruments basically aims to be a one-stop shop for both production and characterization needs when it comes to electrospun fibers. And so in addition to providing equipment that enables the really precise process control and very consistent production of these nanofibers in the form of the BioInesia fluid and tech electrospinners, we also offer a desktop scanning electron microscope called the Phenom, which can be used to quantitatively characterize these fabrics. This desktop electron microscopy is actually probably a really important tool to have in the toolkit when you're looking at nanofibers, critically on account of the fact that the fiber diameters are approaching to, if not below, the diffraction limit of light. So the additional resolution that comes about from using electrons is absolutely required. And there's some unique attributes to the phenom specifically that really make it a great candidate for quality and process control when producing these fibers, as well as an R&D tool for investigating appropriate process parameters in the first place. Okay, so we offer two different models of phenom, both the Phenom Pro X and the Phenom XL. What I'm talking about today is principally the XL. Both the Pro X and the XL are built on a unified column design, so the only real critical difference between the two instruments now is the stage size. And the XL can actually load a sample up to 100 by 100 millimeters, so 4 by 4 inches, or it can load multiple small samples, which is really good in a quality or process control type environment. All phenoms produce significantly superior images when compared to other desktop SEMs, owing mainly to the high brightness cerium hexaboride source that we use, which I'll discuss in a little bit more detail on the next slide. They're also effectively the highest productivity SEM on the market, mainly due to the fact that they have an extremely fast pump downs, 45 seconds. And also, they have a very efficient workflow. There's integrated navigation cameras, so you don't get lost inside the tool. And basically standard integrated motorized staging, which can be automated. Just from a workflow perspective to illustrate, you know, in about 5 minutes with an automated acquisition, you can put the sample into the SEM, pull vacuum on it, and acquire about 40 images. And by contrast to any other SEM on the market, right, which would still be pumping down the sample chamber, that's a huge difference. So you know, we're talking about one image every 5 seconds in an automated workflow, and you can collect hundreds and hundreds of images in an hour, really, without any challenge. So it really is the fastest and most efficient SEM on the market on account of that. All right, so the phenom comes equipped with a cerium hexaboride source, which I mentioned on the previous slide. This is a pretty advanced source. It is offered as an upgrade on a lot of floor model systems, principally because the images that it produces are significantly better than tungsten hair pit emitters, like you see on the bottom portion of this slide. Cerium hexaboride is about 10 times brighter than tungsten, which gives us a broader accessible contrast range in our images, which can be extremely powerful from a post-processing standpoint. The lifetime is much higher, which minimizes the amount of maintenance the instrument needs. So 1,500 hours is kind of a minimum lifetime for our sources, whereas a tungsten emitter, which generally get about 100 hours of use. So there's significantly less downtime on our system. Another benefit of cerium hexaboride specifically is that it emits electrons about 1,000 degrees C cooler than tungsten guns, which means that they have a narrower energy distribution leaving the source, which enables us to focus them more tightly, particularly over a short run. And so this source type really is the driving factor for the miniaturization of the SEM. And yeah, without it, frankly, the resolution just would not be as good. Probably would be different by an order of magnitude. The source does not fail catastrophically, so it's not like a light bulb filament that burns out every 100 hours. It fails gradually, and usually after that 150-hour mark. And it's much more stable, mainly due to its increased size, and so you don't get issues with stigmation and tilt having to be readjusted constantly. We use generally backscatter electrons, and we'll talk about that a lot when it comes to this particular talk. Backscattered electrons are elastically scattered, so they have a higher energy leaving the sample. And the big benefit for the nonwovens market essentially is that the air in that chamber that we can leak in basically some additional air into the chamber, which is nominally ionizable and will actually act as a conductive path to ground in most instances. So if you use a backscatter detector, you don't necessarily need to gold coat samples, which minimizes sample prep pretty drastically and also eliminates the potential for some artifacts to be put into your image. Another benefit to backscatter detectors is that the image contrast and brightness depend on both the topography and also the atomic number of the analyte. So the higher the atomic number of the surface, the brighter it will appear in the overall image, which can be used to really quickly identify foreign particles and things like that. Here's the geometry of the detector in the phenom. All right, so the detector is an annular detector, and the beam actually comes down through the center of it. The image is reconstructed on 4 separate quadrants, and the really big benefit to using this type of geometry really is that the light source, right, those electrons, and the camera, basically the image is reconstructed as if those two things are coming from the same point. And so we don't see any shadows. In our SEM image, the shadows are basically cast backwards into the image as opposed to being at an angle or something like that. And that drastically improves the capacity to post-process these images. So on the left-hand side here, I show a backscatter image of a HEPA filter, right? Again, showing basically no shading associated with the orientation of the detector with respect to the sample. And on the right-hand side, I show a secondary electron image. And because the secondary electron detector is mounted sort of at about 10 o'clock here with respect to this image, you see things like this fiber down here has a bright side and a dark side that corresponds to the orientation of the detector with respect to that fiber. That's really nice for adding depth, you know, to the image and really producing like pretty images generally. But it makes quantitative post-processing extremely challenging, right? Because I can't threshold this image into a binary image and have it look good, you know. So here if I do the same thresholding methodology on both images, I get pretty clearly defined fibers on the backscatter, again, owing to the fact that the shading is uniform across the fiber. And on the secondary, I get something that really would be impossible to work with. Probably the fibers that are properly found or have the potential of being half the size on account of the bright edge or something like that too. So post-process, as secondary images are really great for looking at, and you know, they give you some you know, pretty depth and good coloring information sometimes, but the backscatter is really sort of necessary for post-processing. So just as an application example, we have some electrospun pan fibers here, and I wrote a Python script to control our stage and basically had it acquire 10 images at random. Again, this is about 5 seconds per image, and I was able to autofocus and maintain contrast and brightness across the whole run, and the total acquisition time wound up being just shy of 1 minute. So we can acquire 10 images really quickly inside the SEM. These images were then fed into our fibermetric software, where they were effectively thresholded down into the individual binary image showing the individual fibers. And then the software identifies all of the center lines of these fibers, and then we'll randomly pick a user-defined number of center line positions to make measurements. And so the user-defined number in the case of this experiment was 100 fibers per image. So there will be 1,000 total fibers measured. And now in under, I think, 4 minutes, we've produced basically a report with data that can be exported into Excel, where 10 SEM images were measured, 1,000 total fibers were identified and measured, and you wind up with a nice histogram showing both the average and median fiber diameters to be somewhere around 340 or so nanometers. This is exceptionally powerful because there really is minimal operator bias, right? We've done gauge R&R studies on things like melt-blown fibers where the diameter distribution is quite broad, and you can find from operator to operator something like 20–25% variance in fibers measured, mainly on account of the fact that there's a very low number of fibers measured in total, and because of the fact that, you know, John might really like the big fibers and Mary might really like the small fibers, and so on account of that you wind up with funny distributions. Fibermetric removes all of that operator variance, which is really key, and produces both accurate and very precise data. These pan fibers were actually spun on a slow-moving drum, and so one thing that's kind of interesting to note is that the fibers themselves look pretty randomly distributed, but if you actually plot the histogram of their orientations, you do see a pretty clear preference towards what is most likely the machine direction and the cross-machine direction. And so you can use data like this to solve process problems or R&D problems, basically, as you're producing these fibers with the fluid in the tech. And you can go back and tune either the drum, the feed rate on the drum, or the distance between the emitter and the collector, in order to better optimize these orientations. This is kind of a neat experiment as well. So this is air filter, pulsed air filter, with electrospun PVDF on the surface of it. I think this is a typo. The underlying substrate's probably polypropylene or something like that. But one thing that's pretty clear here is that there were issues with shot particle formation from the electrospinning apparatus that the customer was using. So the viscosity was probably wrong, such that a droplet would form at the end of the needle and shoot this particle out occasionally. There is a web there that you can barely see in some areas. But what I did basically was I turned the voltage up on the SEM pretty high to 15 kilovolts. And at that level, fibers under 100 or 200 nanometers actually become mainly transparent. And so all you see are these shot particles suspended over top of the underlying substrate. You can then threshold this image and quantify basically particle shape, size distribution, frequency, nearest neighbor separation, coverage. All these numbers can be teased out of this image and fed back in again to more precisely control the process parameters. And here, you know, it probably would be the control of either the viscosity of the solution or the temperature and relative humidity of the instrument that is doing the electrospinning that would really ultimately serve to remove these particles. So the phenom can be really powerful for solving these types of problems as well. Lastly, but not least, all the phenom SEMs can be also equipped with a EDS detector. So this is basically an energy dispersive X-ray spectrophotometer that you can use to basically use the electron beam to excite X-rays from a location in the image. Basically, every element on the periodic table produces a unique signature of X-rays, and so you can use that X-ray spectra to quantify the elemental composition of what's on the surface. This is not. It's important to note this is not chemical information, so I can't tell the difference between, you know, polystyrene and various polyethers, for instance, because they're all carbon and hydrogen. But you can delineate the difference between foreign particles and things like that on the surface, and figure out, you know, is it a steel flake? Is it part of my nozzle? Or are there foreign contaminants coming into the system? And tease out that information pretty quickly. In addition to the electrospinning equipment specifically, it's important to note that basically all of the image processing information that I showed, along with the SEM, can be adapted to a huge variety of industries, ranging from nonwovens to filtration. Again, here's an electrospun nanofiber on top of, in this case, it is a paper backing, and here the voltage is more carefully optimized to visualize the fibers themselves. You could look at flock fabrics, textiles, paper fibers, membranes. When you have two-dimensional membranes like this, actually we can use the lack of shading again to do things like quantifies the pore size distributions in these membranes, as well as the strut, I guess they'd call it, thickness in between pores. For paper, we've done some interesting projects looking at basically the quantity of precipitated crystals used as brighteners and things like that on the surface as well. So it really is a broadly applicable tool to this industry at large, and really great for quantifying shape and size distributions of nanofibers, orientation information about nanofibers, particulate coverage, pore size, et cetera. And again, it does it with the highest throughput of any SEM on the market. So in conclusion, effectively, the Fluid-in-the-Tech line is basically uniquely precisioned for precision generation of high quality product. It is basically the only commercially available electrospinning apparatus that has extremely precise humidity and temperature control, which is critically important for particularly any sort of production type project where you need very uniform product, and it needs to be uniform a week from now as well. So that's really great. And then coupled with the phenom, it's a great R&D tool for optimizing process parameters for the Fluid-in-the-Tech line, and generating really great images very quickly of the nanofiber membranes, I think.
All right. Thank you, Matt, and thank you, Tom, for your wonderful presentations, and thank you to all of our attendees and your wonderful questions. We will be responding to those offline, and I apologize that we did not get to everybody's questions. I was trying to keep to our timetable. Again, you can visit us at the filtration exhibit or the BMES show in October. We'll be at both of those shows and featuring both the phenom and the Fluid-in-the-Tech line. You can contact us through any of those emails. Our phone is listed there. Our toll-free number is there. You can find out more information about Bionesia on their web or about nanoscience on the web. And if you are on any social media platform, LinkedIn, Facebook, Twitter, or Instagram, please follow us on there. We will be sending out the recording of this presentation to everybody that was on the attendee list. Thank you again, and have a great day.